Bonjour tout le monde, bienvenue à ce quatrième webinaire, puis avec le background, ça va mieux aller. Je vous souhaite une bonne année 2026 à tous et à toutes. J'espère que vous allez bien. Donc, on commence l'année en force aujourd'hui avec vraiment une belle présentation. Ça va être vraiment intéressant. On reçoit un invité qui, en fait, va faire sa présentation en anglais. Donc, je dois le dire de prime abord, là. L'échange de la présentation va être fait en anglais aujourd'hui. C'est possible quand même de poser vos questions en français, puis je vais me charger de les traduire là, sans problème. Vous me connaissez, ça fait quand même plusieurs présentations que vous me voyez. Donc, Jonathan Pratt, je suis le directeur général du RQMO. Et en fait, aujourd'hui, on reçoit pour la présentation professeur Paul Dutchak de l'Université Laval. Hi Paul, how is it going?
Very well, thank you. Thank you for the invite.
Well, thank you for accepting this invitation. It's really nice that we can have a presentation in English. We know that a lot of the research are usually done in English, so it's kind of I'm very happy to have you with us today. Can you tell a bit more about yourself? What are your research topics and what are you working on?
So I actually came here to the University of Laval just a little bit before the pandemic, where I started my lab to do some research. And we are kind of progressing with some of the research that I started as a postdoctoral researcher at the University of Texas Southwestern Medical Center in Dallas, Texas. And so through that pathway, right now we are looking at some of the important genetic regulation that's behind certain neurological diseases, kind of encompassing epilepsy and autism spectrum disorders.
Okay, great. So you're a professor, you're an assistant professor at the Université Laval?
I am an associate professor right now.
Associate professor, so it's
a middle rank. Rank.
Yeah. Okay, great. So, let's jump in the presentation. I'll just move you ahead of me because it will be easier to follow. So, again, if you have any questions, si vous avez des questions durant la présentation, vous pouvez les mettre dans les commentaires, que ce soit sur Facebook, LinkedIn ou YouTube ou directement sur StreamYard. On va répondre aux questions à la fin, donc je vais me charger de les traduire. So, we'll answer the questions at the end of the presentation. So, Paul, the floor is yours.
Excellent. So thank you very much, Jonathan, and thank you to all the organizers from the Rare.qc program. I think it's a really fantastic program for us in Quebec, and I'm really happy to see that the efforts being made to put forth the importance of some of these rare diseases and really try to expand science beyond the classic genes and diseases that we all are mostly told about. And so today, what I'm going to talk about is a little bit of our program, really looking at nutrient signaling pathways that control how cells function, right? Because we understand that, it's not only just our genetics that are in play in what make us who we are, but it's also what we eat and how our body uses those metabolites and things to develop and function accordingly. And so the title of my talk is when nutrient signaling goes wrong, how metabolic signals can promote epilepsy and autism. And again, I'm from the Université Laval, where my research is really focused at the Centre de Research, CERVO. So just to start with a bit of an overview, I just wanted to kind of bring in some basic biology into this talk and just kind of mention that, our brains really function by the coordinated interactions of multiple cell types. These can be neurons or astrocytes and glia. So all these different cells within the brain, which are organized primarily into circuits or pathways. And it's really the communication between these different cell types that allow us to think and give us our neurological function that we depend on as humans. The problem is that sometimes these circuits don't develop properly, or they coordinate poorly, and really these functional differences are what can cause circuits to become amplified or produce pathological states. And really in our lab, we're looking at primarily seizures as an output of these dysfunctions, because I think, you know, we do a lot of research in the lab using animal models. And so seizure, you know, animals having a seizure, it's very easy to visualize, and it's not so much of a behavioral phenotype that we're looking at. They're very strong things to see. And so our research is kind of focused on this intersection between two neurological diseases, one being epilepsy and the other being autism spectrum disorder. And I think while these are generally viewed as very distinct, you know, diseases, disorders, I think the genetics that are in play right now are really showing there's a lot of intersection between both. And it's kind of causing us to re-examine our definitions of, you know, what is epilepsy and what is this autism spectrum disorder. And being a spectrum, there's a variety of different, you know, factors involved with that. But just looking at the numbers generally, you know, in epilepsy, we generally say that there's around 300,000 Canadians that suffer from epilepsy, which is a big population. It's not really a rare disease per se, but when we start to dissect, you know, what are the actual components in the different types of epilepsy, it becomes pretty clear that there's certain ones that are more rare than others. And again, with epilepsy, which is affecting about 1 in 66 children in Canada right now, again, it's prevalent, but we're starting to understand, you know, what are the factors that are actually causing this, and why this spectrum of disorders. And so in my lab, we are really looking for the strongest intersections on some of these diseases, and really trying to boil it down to like what genes are dysfunctional, and how are they actually contributing to these different types of neurological diseases that are generally considered distinct, but really, you know, at a biochemical level are not. And so just one interesting point I wanted to raise was that approximately 30% of individuals with autism spectrum disorder experience seizures. And so it's not really completely clear why this happens, but I think it's an avenue that we're interested to investigate and see whether or not we can actually limit that type of outcome. And so, you know, the first question we consider is like, why do seizures occur to begin with? And so seizures can occur from a variety of different mechanisms. They can be either genetic, you know, you're born with them, or a mutation that can occur, or they can be through an acquired situation. So genetically, classically, these are usually considered to be these classical channelopathies, where there's mutations in genes that are important for coding specific proteins that function as ion transporters, and really are important for controlling how the neurons, the electrical responses of the neurons themselves. And so that's kind of easy to visualize how that might influence seizure biology. There's a lot of other different types of conditions that can cause seizures, like these acquired conditions like trauma, stroke, formation of brain tumors can elicit tumors, as well as metabolic alterations or other diseases. You know, for instance, with diabetics, if they have a hypoglycemic event, they can actually suffer from a seizure as well. And so there's this really important relationship between our genetics and also our metabolism and into how our brain can function and maintain itself. But despite these diverse, you know, factors, they really all converge on the stability or the circuit instability of the brain that's tripping these seizures off. And so I think from our point of view, we don't really necessarily view the brain as this beautiful, I'm not sure if you can see my arrow or anything, but this beautiful infrastructure that allows us to think and do all these marvelous things as humans and other organisms as well, or the cellular levels of how these cells interact with each other. But in our lab, we're mostly interested in really the molecular, the biochemical pathways that are actually behind this whole process. And as we know in the lab, these are very intricate processes that allow us to actually have brain functions. And so we are looking mostly at the cell biology here, and also the factors like genetics, proteins, and metabolites that are important for coordinating with the biological function of the cells to ultimately mediate, healthy brain activity. And so in the realm of science that we study, with epilepsy and autism spectrum disorders, there's been a real surge in advancements, really that came out of some of the genomic sequencing studies over the last several years, and the development of these DNA sequencing techniques. And as you can see here, with the epilepsy-linked genes, originally epilepsy was really considered a channelopathy, a mutation in some transporter or something early on. But in the early 2000s here, with the development of these DNA sequencing approaches and genomics as a field, we've really begun to just identify a variety of new mutations in different genes in different people that are also contributing to this type of neurological disorder. Same thing goes with autism spectrum disorders, and people have been looking at what genes are in play and what types of mutations and how do they affect the severity of these diseases. And so in our lab, we like to look at intersections of genes and how genes function together and in the context of biology and where they go wrong. And so if we start looking at the overlapping genes, a lot of them, yes, are in ion channels, but there's unique aspects, including mTORC1 signaling, which is an intracellular signaling pathway that we're going to talk about a little bit more later. There are other things that regulate like gene expression at the chromatin level, or also neurodevelopmental or scaffolding proteins. And so, it's really nice to have all of these genetic links, but in a way, what they represent is part of the blueprint of what makes us humans or life living. And so knowing those little details, in biology help us, but we still need to figure out, what is the function of these genes? What are the function of the proteins that they code? How does it work in the biology of the system? And so that's, what I really like to do is kind of a scientific sleuth in a way, trying to figure out how are these genes working and how do their dysfunctions contribute to the disease. So getting a little bit more specific into our research, there has been a nice convergence of some of the genetic clues and some of the molecular insights over the last few years, with these genomic sequencing studies being mapped onto some of the genetics that we've been looking at in our lab. Namely this particular protein complex called GATOR1, which is a complex that's comprised of 3 different proteins. And so, as you might imagine, our DNA makes mRNAs, and then the mRNA makes protein, and some complexes in our body require multiple proteins to come together to function properly, right? And so this early study in 2016 was really the first to really identify and characterize new mutations in a big cohort of people that had epilepsy, and they have identified that this complex called GATOR1 was mutated in these people with epilepsy, which was kind of went against what was generally considered known in epilepsy, because it's a totally different mechanism than the classical models of ion transport and that type of thing. And so during my postdoc, I was actually starting to work on these genes from a different perspective. But with these papers coming out showing these links to epilepsy, we've really begun to focus on, that and, what's causing it and what are the cellular defects. And beyond this initial paper that came out, there's been others now identifying that these genes themselves are mutated in people with autism spectrum disorder. And so I think that's this little link that we're studying right now in our lab. And so one of the strong models that we have developed in our lab, is using animal models based on a genetic change that we made in the animal line that we're studying, where we've created a conditional knockout mouse for one of these genes that make this GATOR1 complex. And the gene that we're particularly interested in is called NPRL2. And so it's a key regulator of this complex function itself. And so using our animal models, we're able to kind of ask some questions about what these genes are doing in the brain, and how do their mutations ultimately cause seizures and these neurological disorders. And so I just wanted to zoom in a little bit more into the cell signaling pathway that we're looking at. And so what we're looking at is really this. It's an evolutionary conserved pathway that signals inside of cells. And it's kind of the two components that we're looking at is this GATOR1 complex, which is comprised of NPRL2, NPRL3, and DEPDC5. And its function as a main regulator of mTORC1. And so this pathway is actually conserved across life, in the sense that it's found in simple organisms from yeast to all the way up to humans. And so it's we really view it as a really important regulator of mTORC1 function. And what it does is it responds to amino acids like leucine and arginine, which are amino acids which we should know make proteins in the cells. And it coordinates the activity of a master protein kinase called GATOR1. And so GATOR1 is generally considered a master regulator of cellular growth. It's often associated with cancer in people because many types of cancers actually have an upregulation of mTORC1 function. But for some reason, when this happens in the brain and neurons or whatever, we're getting some really bizarre electrical activity and different brain disorders that are associated with it. So this complex of mTORC1 is regulated, as I say, by GATOR1 signaling, which is downstream of specific amino acids, as well as growth factors like your classic growth factors, insulin and those kinds of things that signal into the cell. This pathway is really important biologically speaking because it's a main regulator of cellular protein expression or this process we call translation. It's involved in the regulation of degradation of proteins or organelles that are not really being used at the time in a process called autophagy. And through its activity or inactivity, it seems to be able to control metabolic homeostasis a little bit by modulating how the cells really use metabolism of carbohydrates and amino acids and even nucleic acids. So it's a real master coordinator of cells, and ultimately it controls cell growth. But what we find and what we know genetically is that when there are mutations in the regulators of this mTORC1 itself, it becomes hyperactive, leading to disease. And so I just wanted to show a little bit of data from what we do in the lab. And I just wanted to kind of go over the key here, which is that for understanding is that in our cells, when we have high amino acids, so like if you imagine after we eat, we have a lot of nutrients that our body absorbs. In that condition, this GATOR1 complex is retained in an off situation. And by GATOR1 being off, we have this activation of mTOR where the cells are able to use those nutrients and grow appropriately. In conditions where there's low amino acids or starvation, the function of GATOR1 is to activate and then turn off mTOR signaling. So it's a little bit of an on-off switch in a way. But these mutations that we find in people ultimately affect how GATOR1 is functioning to control the activity of mTORC1. And so what we did, last year, or I guess 2024, we published this paper where we actually looked at some of the human mutations in this subunit of GATOR1 called NPRL2 that we put in. And what we find is that there are certain mutations in the human population that actually are complete loss of function. So if you have a specific mutation, that whole complex doesn't work properly, and it is unable to turn off mTOR signaling, which we are looking at here as a surrogate of phospho S6 kinase. When GATOR1 is mutated, we don't get that inhibition that we should when cells are starved. And so in this case, we're using just a basic saline solution that we're growing our cells in, but the cells still think that they have all the nutrients that they need to grow in that. And so our one of the main questions is that we have in our lab is like, is this important, for neurons in the brain? And so a lot of our early studies, we just use basic cell lines and things like that to characterize some of the biochemistry. But when I started my lab, we decided to use our mouse and knock out this gene and this GATOR1 function in neurons to see exactly what was going on behind this. And so what we found by analyzing that model was that when we deleted NPRL2 and caused GATOR1 defect in those neurons, the mice died quite rapidly after birth. So they all died before 20 days after birth. And so, I see my slide is a little weird. But anyway, these mice ultimately all showed signs of seizures, which we quantitated here, which is unfortunately behind some of my other data with this presentation. But they had seizures that were quantifiable, and they all died. And so through that process, we were able to investigate what was going on a little bit at the molecular level inside the brains. And just to show what we see here, these are images of the brains of the hippocampus and the cortex regions, where we can see some of the markers of mTORC1 signaling being upregulated. So when we knock out this inhibitor of the pathway, we see this strong activation that's causing seizures and causing early death in our animals. And so, scientifically, there could be multiple reasons that could explain this. And so one of the questions we had is like, could this be occurring in a cell autonomous manner? Like, is it a defect exclusive to the changes that are going on in the neurons themselves, or is it something else? And so what we're able to do is we were able to isolate primary neuron cultures from the mouse brain, and we could put them onto little dishes, I guess, in the lab that have electrical sensors on the bottom. They're wired to kind of detect the activity of those neurons as the neurons grow in culture over time. And so using this model, we were able to find that over a 21-day time course, that those neurons developed to be very, very hyperactive. And just to kind of show you, here are the readings from this type of an experiment, where we can see early on in this process that by day 14, the neurons start to become activated in cell culture, and we can see this happening. But by 21 days, when the neurons are fully mature, the top being the control or wild type or normal condition, we have this kind of nice routine firing of the neurons in the dish. But when we create these GATOR1 defects by knocking out NPRL2, those cultures become extraordinarily hyperactive and fire almost as you'd imagine what would be happening in a brain that's having a seizure. So it's kind of a neat model that we've been able to find and really show this link between this intracellular regulator to actually a neurological expression phenotype. And so one of the things that we kind of questioned is like, how can the neurons themselves on a dish maintain such rapid communications without becoming depleted? And so, this is a kind of a balancing act, right? Because when we consider neural activation, those neurons themselves are using their energy, they're using their metabolites, they're using their resources, but for some reason these are not becoming depleted, and even in the animal models. So we started to consider a little bit more of like, could these mutations be affecting other cell types beyond the neurons themselves? And so there's been a lot of research over the last many decades that have looked at kind of these interplays between neurons and other cell types in the brain, because we understand that neurons, they're not an exclusive cell that functions in a bubble. They're part of an integrated system. And so one of the important cell types that are in the brain that helps the neurons with their stability and providing nutrients is these astrocytes. And so I just pulled out an image from a textbook, pretty much, that describes the flux of metabolites between astrocytes and neurons. And so we kind of imagine that perhaps astrocytes could be a major contributor to this biological process causing seizures. And so what we did using our animal models is we knocked out the function of this GATOR1 in astrocytes in the brain in the mice, and we asked the question, is the contribution of GATOR1 in astrocytes sufficient to drive these seizures and neurological changes? And so with these mice, when we started analyzing them, we also noticed that they too started to die early. So with the average death point around 200 days after birth, just in the context that most mouse models will, if they're normal, they should live at least a couple years. So this is a early death in that regard. And that by 5 months of age, these mice started also suffering from seizures as well. And so this was kind of a unique and very new discovery. We actually have a manuscript that we have in revision for one of the scientific journals to describe this phenotype. And so we've been really interested in trying to dissect, how is this actually happening? And so what we did was we looked at the brains of these animals at 5 months of age, really when we first start to see these really bigger seizures happening. And looking at those astrocytes themselves, we could see that that compared to control astrocytes, we could see that the knockouts really developed this kind of angry looking phenotype, which is really reminiscent of them becoming hyperactive and reactive within the brain. We've gone on to further analyze what genetic expression differences were occurring in this type of phenotype. And one of the interesting genes that we found to be massively upregulated, which like over 100-fold upregulated, is this particular gene called glutamine synthetase, which is the main enzyme that's responsible for making glutamine in the brain. And that glutamine goes in to supply the neurons for their source of glutamate, which is used as an excitatory neurotransmitter. And so that was kind of the model that we developed. And so what we did just to kind of enhance that, our data to show that, what we did was we actually took astrocytes and we cultured them in a regular, environment in dishes in the lab. And what we did over a period of time is we collected the media kind of to assess, what is going out of the cells into the matrix that could be influencing the neurons, even though these are exclusively the astrocyte cells. And what we find is that in those knockout cells where GATOR1 is defective, they have higher levels of lactate and higher levels of glutamine, which matches with the genetic expression that we're seeing. And so what we kind of imagine is that with these GATOR1 pathologies is that there's this kind of hyperactivation of these neural pathways, both within the neurons and also the supporting cells that are providing, the metabolites and the fuel, let's say, to make the system go. And so the problem with that is that, it becomes a multiple cell type problem. And because these different cells are coordinating to cause the disease. And so, building this in our lab, these ideas, what we kind of thought about is like, okay, can we really come up with a different strategy to target and to treat this condition? Because unlike some of the classic versions of epilepsy, where you have a specific mutation in an ion channel, well, we have different inhibitor drugs that can target those, but those types of proteins are exclusively found on neurons. Whereas in this type of condition, we have kind of an interplay between different cell types that are going on. And so one of the interesting groups of papers that came out during the pandemic era was that a couple other groups had developed these animal models of GATOR1 deficiency and had actually begun treating them with small molecule inhibitors of mTORC1 or little drugs. And what they found in their studies in the animals is that they could actually correct some of the neurological defects that were presenting in their models by treating with mTORC1 inhibitor drugs, which are really these small molecule classic drug systems that binds to this mTORC1 protein complex and turns it off essentially. And for us, this was kind of an interesting avenue. Our research program had definitely shifted gears a little bit during the pandemic. And so instead of following the path of, the well-beaten path of discovery, we decided to go off on a bit of a different idea and see if we could develop a different strategy to actually be able to ameliorate or improve the condition of these GATOR1 pathologies in a different manner. And so you get one of the key new types of molecules that are being developed for rare diseases are called antisense oligonucleotide molecules. And what they are, generally like a small molecule that's like a nucleotide sequence. So, we have nucleotides in our DNA and our mRNA. And so in the lab and through collaborations, we can design these types of these molecules to target specific mRNAs and affect their function. And so there are different mechanisms that people can do this. You can either target the ASO, these types of molecules, to the mRNA, which leads to their degradation. So effectively thinking that you can like turn off the gene or dampen the expression of proteins or signaling pathways. Or there are different approaches that can alter the splicing of genes, which gets a little bit more complicated, but effectively you can change the function of the gene's program by affecting the splicing of exons and introns within those targets. And so this approach is really relatively novel, but a lot of pharmaceutical companies and researchers are pursuing them because it allows us to target particular genes that otherwise we cannot really target pharmacologically, because many proteins don't have like a catalytic function or whatnot. And so by affecting the mRNA level and subsequently the proteins that's produced by these mRNAs or genes, we can actually kind of change the activity of these pathways in a totally new pharmacological manner, which is really exciting, I think, and especially for a lot of these neurological disorders that have not been druggable for many years. And so just kind of imagining our pathway where we're looking at these mutations in the GATOR1 complex that lead to a hyperactivation of mTORC1 signaling, we decided to see if we could design some of these ASO molecules and effectively take a situation where we have an F1 race car driving through the streets of Quebec, which is, we can imagine that not to be a very safe situation, to kind of turn down the activity of this signaling cascade into more of a reasonable paradigm. And so we were able to do this with help of one of my students in the lab. We designed a variety of these different drugs and screened them. And we found a couple that were able to turn down the expression of this mTORC1 protein. And so, realistically thinking about this, if we have less of the protein there, we should have less of a function of it, right? In the cell, it's just the mass action effect. And so after we designed these types of ASOs, we asked the question, like, can this new molecular approach be biologically beneficial? And so we had already developed a couple models in the lab to look at this. And so we started testing our ASO to mediate mTORC1 function. And so one of the models that we first tested were with these primary neuron cultures, where we took the neurons out of our mice, knocked out the GATOR1 subunit NPRL2, and allowed them to grow up on these little dishes. And we were measuring the activity of these neurons over time. And what this data really shows is that over time, as the neurons mature, you can see in this dark purple, the NPRL2 knockout cultures have very high firing rates, which is consistent with what I showed earlier. And when we treated these particular cultures with this ASO molecule, we're able to actually turn it down to like wild type normal levels, which is just absolutely incredibly excellent, I think, and promising. And so moving beyond our, cells in a dish model, we went back to our animal model, which I showed earlier. These are the neuron specific knockouts where the animals end up having seizures and dying very early, about 20 days after birth. And so we asked the question, if this ASO could rescue that phenotype. And so we just tried a really basic kind of experiment. We do know from other literature that these ASO molecules are extraordinarily stable in vivo, and they can last. I think the human studies really suggest that they can last between 8 to 12 weeks per dose, which is a lot longer than a few hours like these other small molecule drugs can do. And so what we did was we just took our animal models and we treated them. We did a injection into their brain with our ASO. And this was the first data that we received. We could see a massive time extension in their survival. And during this time, we did video recordings, and we found that it did reduce their seizures. But we did notice that just prior to them succumbing, the seizures started to come back. So I think there's probably some drug wearing off effect going on that allows this to happen. And so this is what we're actively studying right now in the lab, and I think it gives us some good promise that at least for one of these rare diseases, we might have something interesting for the future pretty quick. And so in summary, I just wanted to go over that I think that the importance of genomic or DNA sequencing studies, which are identifying genetic changes associated with epilepsy and autism spectrum disorders. These are very, genomics is a really important component for science in general, because it helps us identify what the blueprints are and gives scientists and researchers like myself that blueprint to kind of work on and be able to dissect to figure out the biology that's behind it. The second point is mutations in these upstream regulators of mTORC1 lead to hyperactive communications in the neurons in mice, causing seizures, and really define a new class of pathological seizures that I think is really going to be one of these intersections between epilepsy and these seizures that are occurring with people with autism as well. It might be more complex, but I think until we see that, we're gonna think that there's this overlap. And our work establishes GATOR1 dependent seizures can emerge from different downstream mechanisms. And so I think that's an important concept to incorporate into these rare diseases, because a lot of times when we have a disease or a genetic mutation in one cell type, it might actually be doing something very different or unique to that particular type of cell, as we can see in this example. And so I think this really just emphasizes the importance of really being able to dissect these rare diseases into their actual mechanisms of what is causing the underlying conditions, because when we understand the mechanisms, we will understand strategies to ameliorate them, to make them better. And finally, I just wanted to indicate our first in class ASO mediated approach to dampen mTORC1 activity can ameliorate pathological effects of GATOR1 deficiencies across multiple cellular and animal models. And so this is where we're really actively pursuing our research right now, with the hopes of maybe bringing this to a clinic in the future day. And I think with these ASOs, I just want to also leave us on a promise, on a bit of a hopeful note. These ASOs are really providing new hope for a lot of neurological diseases. And some of you may have seen this story from CBC, from my home province of Saskatchewan, which really details one of the success stories with ASO in the field of ALS or SLA, or Lou Gehrig's disease, which researchers have now developed ASOs to treat a particular variant of ALS. And this is an example with Paula Trefiak, who is taking this ASO and has actually totally block the progression of ALS, which is a devastating neurological disease that has had no therapeutic hope in the past, but she's now stabilized and actually has regained some function of the loss that she had with that disease progress. So I think, with these types of new therapeutic approaches and support from, groups like Rare.qc, us scientists are really feeling, excited that we can actually move forward on a lot of these conditions that, previously weren't considered treatable. But I think there's new tools that are opening up new avenues. So something to look forward to. And with that, I'd like to acknowledge my lab members at CERVO, where my main lab is. It's off the campus of the University of Laval. But I have a small lab with Maylene, Iman, Antoine, and Joey as my PhD students, and an excellent research technician, Leticia. I have a few different collaborators, Dr. Chantal Sefton, Marc Etienne Hout, Martin Levesque, and Andy Nguyen at University of St. Louis. And I'd also like to thank my research sponsors and thank all the groups that have helped support our research endeavors, all listed in the corner here. That's everything for now. So if anyone has any questions.
Yeah, thank you so much, Paul. It's I will put the camera like that. It was so interesting, and actually I have one question and one observation, but we'll go into the comments. For now, we only have one comment from Lou Bella, or Ayu Bella. No, Lou Bella. Yeah. So very promising, très prometteur. Merci beaucoup et bonne continuation. So she's actually very happy, or well, they actually are very happy, because so sometimes the username can be tricky. So, but thank you so much for being with us, for being with us, and thank you for the comment. Actually, we have another comment from Lilith Antonian. So thank you for the great presentation. Do you envision that the ASO therapy for the gene that you study will be given to patients only intrathecally, or also systemically through an intravenous manner? So that's the first question. And the second one will be, what do you think of other delivery methods, like intranasally, so through the nose?
Yeah, absolutely. So right now, it's a complicated question because there's many angles on it. But I think for us right now, with the neurological conditions, we are thinking in line with what people are doing with ALS in that regard. So a lot of it's intrathecal injection. The interesting aspect that we are also considering for the future is that these mTOR hyperactivity relationships with cancer, kidney function, polycystic kidney disease. There's different mTOR related pathologies outside of the brain as well. And so that is something that we are really considering right now. I think, for the neurological aspects, intrathecal is probably the way to go. But there's different strategies to deliver these types of things, and it's an active thing that we're looking at.
Great. Thank you, Lilith, for your question. And I will start with my observation, and if you want, Paul, we'll go back in the presentation, and I'll put the slide because I took note. I think it was on slide 12. Yeah. So is it me, and it's the well, I was a researcher also in another life. 10 years ago, I completed my PhD, and I was mainly working on brain cancer, working with MMPs and all that stuff. And my thesis was on autophagy. So I know a bit about mTOR and BCL2 and all that stuff. So but we'll go on that afterwards. So I was looking at the picture, and yeah, I have, in parentheses, I also have to add that I was mainly doing confocal microscopy and everything. So each time there are photos like that, I'm very happy to see that again 10 years after. But Yeah. So is it me or the knockout astrocytes are bigger than the control ones? And that was the first observation I have. And the other one would be, is it also me? But it seems that they have more interaction within each other. Is it possible? Is it or is it just me?
Absolutely. That was what, part of one of the questions I had from one of the reviewers of our paper was whether or not they were just bigger or there were more of them. There's not more of them, but they are bigger, absolutely. And that we call a phenotype, that extension of these processes is a very reminiscent of reactive astrocytes. So when there's damage, if it's a metabolic challenge, or even in some neurodegenerative diseases where there's death and dying in particular regions, astrocytes can kind of flare out like this. And people are looking at why does this happen? Is it protective? Is it part of the pathology? It's not, those answers aren't really clear right now, but it's definitely, it's something that's observed in that consistent kind of stress condition, okay, as it were. And so, yeah, there's some pretty clear, bird's eye perspective changes that are being affected. And so we're trying to dissect out what's going on at that molecular level that's okay, driving that.
Great, because earlier in the presentation, you were saying that you were looking at how are the mechanisms for explaining the seizure and the way that the metabolism is used in a way. So if they are bigger and they can interact more within each other, maybe that provides somehow an answer to how they can have this this high
activity of glutamine.
Yeah, that's it.
Yeah, absolutely. And quite honestly, like our gene expression changes that we're seeing were extraordinary. I mean, I've been studying gene transcription and that mechanism for in my PhD as well, and to see a gene actually being activated, beyond 100-fold, that's a massive genetic change. And so this is it's interesting.
Yeah. And okay, so we don't have any other comments in the comment section, but actually I have maybe one last question, and that's I think it was on slide 18. Yeah. So you were saying that for the you can reverse using the ASO blocking mTORC1. You can somehow reverse the phenotype. So like for a couple of days, actually, you see that there's more survival with the reinversion, if I can say, of the phenotype. But at the end, we see at 55 days that you still have somehow the deaths of these mice. And I remember when I used to be in my PhD, we had a lot of questions regarding the autophagy itself. So the processes of autophagy. So we know that it can be a survival mechanism, but also a death mechanism. So is it something, could it be something related to these? Because we know that, like you said, mTORC1 is a big actor in the autophagy pathways.
Absolutely. It's hard. We've just started to look at the autophagy pathways as well a little bit. What we know is that generally when mTORC1 is always on, it does tend to block the autophagic process in cells, which kind of gets into realms like Parkinson's disease or some of these protein aggregation disorders. But we haven't really looked at it in this context so much. On a cellular level, in regular fibroblasts and things that we study in the lab, we do see defects in autophagy, but whether or not that itself is contributing, it's hard to say. I think the metabolism and the fuel that's driving these neural activity pathways is more important, and autophagy may be a player later on. But I think, it's model and context dependent, because, without any treatments, like these mice die, 20 days after birth. So it's a very finite period. And so, when these things are actually biologically implicated versus, pathologically dysfunctional, it's hard to know. But it could be, it's definitely an important pathway. And I think it might be a factor in contributing to the early lethality, but it's definitely this it's a challenge. I think that's the challenge with a lot of this type of science, is the chicken or the egg, which one came first. So
yeah, of course. And we have one last comment from Christine Yerjo. She said, "Thank you so much for this great presentation," and I have to agree with her. It was a great presentation, well done, and it raised important question for the following the years to come, in order to find a treatment, and it's actually for people who maybe we're asking at the beginning of the presentation. Usually we're focusing on rare disease, and we see that epilepsy and autism seem to not be in this category, but still we can call them orphan disease because they don't have any treatment yet, or the treatments are not that working well. So
that is something that this Rare.qc program can do, is to pull out and actually, dissect these diseases. Like even cancer, 30 years ago, cancer was one thing, right? And I think through science and research, we are knowing now that there are many subclasses, and technically, like every disease can be kind of viewed through that lens when you dissect it to that level, is what are the genetics that are in play. And so this is one of the most fantastic things that I think you guys are doing. And so I'd like to thank you as well for organizing that within the province here. It's great.
Well, thank you so much, and thanks for Rare.qc. Of course, we're great partners. So I don't know if you have any last words, Paul, for today. I know it was a long presentation. Usually we do around 25-30 minutes. Now we're almost an hour, but it was so interesting. So thank you again. So I don't know if you have any last words.
No, thank you very much.
Thank you so much. So merci beaucoup à tout le monde. Le prochain rendez-vous pour la prochaine présentation, pour la 5e en fait présentation de la collaboration RQMO Rare.qc, va être donné par professeur Cynthia Gagnon au mois de février prochain. Donc là-dessus, je vous souhaite encore une fois une bonne année 2026. Merci à tout le monde d'avoir été là. Thank you everyone, and have a very nice day. Thank you, Paul.
Thanks.